Hi everyone, I'm Jaya Vishwanathan, and you've found the No Brainer podcast where I discuss all things neuroscience in delightful conversations with various experts. I hope you enjoy today's episode.
My background, I worked on auditory processing, and I'm very excited, for example, by the speech as a digital biomarker and what insights it might give us. But then, as you said, I completely agree. You have to kind of think about using all these computational techniques like AI to kind of start pulling out the differences. But then there are so many other factors that could influence it, like you had a bad night of sleep and then your speech can be a little bit slurred. And so, is it because of Alzheimer's, or is it because you didn't get enough sleep, or is the lack of sleep also influencing that? So I get it. Makes sense. Yeah. But I guess that's the fun of it too, at some level, for all of these things, right? So one of the things I want to mention, and maybe you can highlight, is I'm always concerned whenever I hear about a test having been approved, or a diagnostic, I don't know, whatever, like a tau value, a threshold, as saying, oh, if you have about this threshold. A lot of those tests are being conducted on primarily Caucasian samples or participant patients, whatever, and that, what are the challenges perhaps in getting maybe wider samples, more people, more inclusive research. But so more inclusive research to have these diagnostic or any kind of biomarkers be more robustly representative for getting to this idea of let's treat someone for what they have. Kind of like a precision medicine approach.
Yeah. That's a great question. I think there's a couple of things. So whether it's in the therapeutic space or in the diagnostic space, both of these suffer from the same sort of problem, and that is that in the early days of both of these fields, which are very closely aligned, by the way, they're basically sister processes. One of the things that has happened is that the bulk of the patient population that served as the reference for what's considered normal was largely young white males, unfortunately, or in some cases they were smart enough to include women in those populations. They were only done largely in people of Caucasian descent, and this is somewhat problematic because, as we know, white males or females don't represent the entire complexity of the biology that exists in our world. In particular, several of the genes that people harbor that they're not aware of actually come from somewhere in their history where family members may have had children from mixed marriages, and so some of those genes came from either the white portion of the family or the family member that was from a different ethnic background, and as a result, those genes then carry on for subsequent generations, and as a result, the standard 20-year-old white male or female may not be indicative of what you see in the general populace. And so this really speaks to several things. One is the need to broader populations, but also to make sure that we have a better understanding of the complexity of the biology as it relates to ethnic and biological makeups, because even people who are of color, they're not just one ethnic background, they're a mixture of all these different life history, and as a result, their genetics carry through. And so for instance, genes in Alzheimer's disease, like ApoE, the late onset Alzheimer's disease gene ApoE, in particular the allele E4, people carry that allele. They're not aware of it, but they carry it. And in some cases, there are other people who carry the protective form of that allele called E2. And what's really interesting is that those people aren't aware of it either. And so, the origins of each of those particular allele sets came from different locations. E4 largely came from out of Africa, and E2 largely came from sort of the northeastern portion of Europe, largely Neanderthal based, relationships between what we think of as Homo sapiens today, and Homo sapiens sapiens bred with Homo sapien Neanderthal, and ultimately generated offspring that carry these genes. So making sure that we're inclusive of a broader population and representation of ethnic backgrounds, sexes, and geographic locations is, I think, super important because this speaks to your point about precision medicine, because whether we're trying to diagnose somebody by using a tool to monitor those things, or we're trying to use a therapy to modify the biology, in each of those cases, we need to understand the underlying biology first, right? And of course, that means you need to understand the factors that are driving that biology. And this includes the genes and their environment and how those interact as you age. So it's a complex interplay between all those different processes.
Yeah.
And
that's wonderful. So thank you so much. And so I think that's a great segue into my next question, which I promised in the beginning, which is to talk more about Alzheimer's. I think the ApoE was a great example. I also read somewhere, and maybe you can, this is definitely a tangent, but that the reason the ApoE4 evolved is to it has a protective effect against infectious diseases to kind of get people through the reproductive age. But then as we age and live past what Homo sapiens originally were living up to, I don't know, 40, 50 years, but now we're living almost double that in some areas. So now that has become a detrimental allele in that context, which I always think is kind of fascinating, and goes to your point of everything has its pros and cons. And so if you think about every gene in your body and every allele of that gene in your body, it evolved for a purpose, maybe based on geography and location to give you a certain advantage. But then if you take it out of that context, it can turn detrimental. But so if you can talk more about Alzheimer's. Sorry, I just got excited about you mentioning ApoE4 and wanted to share that.
Yeah, absolutely. So, it's interesting because, as you just pointed out, it has been thought that was largely to be protective, to stave off infection, as you said, to make it so that you could live long enough to be able to reproduce, of course, right? And then, of course, the Neanderthals carried a totally different allele set. They weren't exposed to those same sort of environmental pressures that ultimately selected for the mutations in the African descending populations that carried that allele. Okay, and so what's really interesting about that is that as we think about the overall process of selecting things or developing therapeutics, start to a different view of the world. I think previous to this, we've largely thought about therapies as single agents or diagnostics as single measurements of a process, right? And these diagnostic systems have evolved so that you have a readout of what's going on, and you consider that particular biomarker, one biomarker at a time. And so what I'm talking about. As an example, in the cardiovascular field, we measured cholesterol for so long, and we've basically picked a magic number that said, cholesterol has to be below this number in order for you to be considered protective. And then we come to find out that it's really not a magic number. It turns out that it's ratios within your own body between the high and the low cholesterol levels, high density and low density cholesterol levels that are important. And so what it turns out is that, these diagnostics and therapeutics have sort of evolved from singular types of therapies to multiple types of therapies. So in the case of the oncology field, for instance, oncology tends to now, and as they're treating cancers, they're not just using a single agent, they're using cocktails of agents. Because we're not trying to kill just a cell and stop it at this time. We're trying to stop a whole series of cells, and including the mutations that might occur that prevent those cells from metastasizing and getting out into the main circulation. So if you think about that as a concept, one of the things that our lab has done in this space is to try, and this is sort of going back to my comparative roots, so it's a little hard to divorce yourself from that, but that's part of who I am and how I think about things.
And
Is
that's
that
wonderful. Yes,
well,
please.
that,
Yeah.
thank you. But one of the things that we think about is that biomarkers and therapeutics are not just singular measurements. We don't want just a single measurement or small clusters of measures. Our lab has a different philosophy about this. We're exploring multimodal biomarkers in concert to generate essentially a fingerprint. And so if you think about that, that kind of makes sense, right? This goes to precision medicine. Each is different in biology. Their biology is going to be very different. Their diet is different. Their sleep patterns are different. Their alcohol use is different. And so what we want to do is we want to establish a series of metrics that allow us to monitor that patient over. So what our lab doing over the past several years is to develop some frameworks that allow us to use these multimodal, as we call it, so multiple input measures to generate the finger how you're progressing in these. And we've developed this and patented this just over the past several years and have integrated it into a series of very simplistic diseases, just as a first foray. So as an example, we used multiple inputs and multiple input streams of data along with the metadata for that particular sort of we know about them. And we applied this to a very specific disease, a disease that has one gene, a mutation in one gene that generates the disease. It's called cystic fibrosis. So the gene is CFTR. There are mutations in that from the basolateral surface into the apical surface, and normally water would follow that and make the fluid along the inside of the lining of the lung maintain high moisture content. And of course, there's also mucus that's being produced there that is now maintained at a nice slippery and fluid level. Well, in the case of cystic fibrosis, that doesn't occur, and as a result, you get thick mucousy accumulation that can't be cleared from the lung, and then that results in the disease. We did apply these kinds of markers, these multimodal biomarkers, along with the metadata, and we applied it to a clinical setting. We went and pulled clinical data that's retrospective and asked the question, how accurately can these multimodal inputs? And surprisingly, with even a limited subsize sample population of around just under 200 subjects, we had an accurate around. And so develop a precision medicine approach like that, which has high accuracy and high predictive validity that you can apply to things AD and AD related, because we think that that's a more complex disease. So we tend to collect a lot of measurements, but currently they're not being used together. They're being used sparsely, independently. We look at amyloid scan and we say, oh, you've got amyloid, or you've got a reduced glucose metabolism, where you are in the trajectory of your disease. But they're not being used for anything other than that. They're just saying, here's where you are. But you might imagine that that fingerprint can also not only be used as a diagnostic. To say, here's where you are. Here's in that particular setting. And so in our case, one of the things we're quite interested in is applying them to a real world situation. And can we predict not only what would be your stage at your current time, but what might be the best course of action to help attenuate the progression of your disease. And that may include doing things like combining therapeutics or therapeutic regimen like ours to use amyloid antibodies. But that's a single therapy, a single target. But many of these people have comorbidity, medical disease, so you might consider combining a drug that you would use for diabetes, something that is really in that base, and that help reduce some of the inflammatory load that is driving some of the processes, and then also help clear some of the amyloid at a stage when you could preserve as many neurons as possible. So this is an idea, an approach that sort of speaks to this precision medicine, and it's something that we're actively on. We have some patented approaches that we're now applying in this space to try and help answer these kinds of questions.
No, that is very exciting. Thank you for sharing that, Paul. Like I'm personally very, very excited to see what's going to come out of your lab from this approach. Thank you. And I didn't know about the cystic fibrosis experiment was 90% successful. So thank you. That's very impressive and promising. So I'm going to shift a little bit and start talking a little bit about your career and maybe some career advice that you have for folks. So you worked on different biological systems. I kind of have two questions. One is regarding, like, do you have specific advice for people? But then I also want to kind of maybe talk about what your approach is in, you've shifted focus and yet maintain scientific productivity. And, how do you manage your time? And, any kind of little tips for, people who are trying to get into the field and work in this kind of a space, who are thinking about it, who might be in high school or college and are thinking about it down the line, but also maybe people who are tuning in who are maybe in their grad studies and kind of thinking about, how do I manage my time better? So, or any of these things better.
Yeah, so we kind of touched on this earlier a little bit. As a classically trained comparative physiologist, I kind of think about problems as a continuum of activities. Right? And so we tend to approach problems in that way. So in the case of, we were talking about earlier in AD and related dementias, it's not simply a single brain disease. It has aspects of cardiovascular and metabolic syndrome and bioenergetics to it. And so I tend to think about problems in that way. They're not just individual silos of activities, but they're one continuous area of research, if you think about it, right?
Yeah.
And so this systems-wide approach allows us to ask and answer questions across a number of diseases and learn from and be informed by those diseases. So for instance, if you think about AD and neurodegenerative processes, these are processes where the brain is losing cells, we're losing cognition as a result of it, and ultimately it results in cell death. And if you contrast that with something like oncology as an example, this is the opposite process. We have unbridled growth, we have immune system participating in these processes to assist the cells to grow by clearing neighboring community cells. And they also are helping by feeding cofactors and proteins to the tumor cell to help facilitate its growth. And so these are really kind of two sides. I think in terms of question regarding productivity and how does one work across a number of different domains, I think it's about how you visualize the world before you. So science really isn't boxes that we work in, and we spend whole activity and are experts in a particular single domain. Instead, the way our lab tends to think about it is that these are just one continuous process. And so if I learn how for instance, for unbridled growth and the cell mechanisms that are allowing that, people to leverage those and learn about how those are downregulated Alzheimer's and try and reverse it by using therapeutics that are also developed for the oncology field, but just applying them in a new context. And so it's just a difference in how one approaches problem solving. It's more inclusive. And one of the things that I tell graduate students and postdocs and research scientists that are in the lab is that you should read beyond your field, go on in fields, because there's learnings that are occurring there, and you might be able to apply that to your particular question of interest. So one of the things, that, as I've mentioned, we develop diagnostics, but that's not all we do. We're developing tools because diagnosis just modify the biology. So if I want to see biology is changing in response, I need some way to visualize that. So they're paired responses.
Yeah.
Much in the same way that we're looking at these paired responses for oncology and Alzheimer's. So it's one large table, a buffet of opportunities to ask and answer important questions and use those to
Yeah.
drive your research. Think about a student that's coming up, I guess, throughout their career, and they're looking to get into this field. I mean, one approach, one traditional approach is, okay, you can pick a particular and become the expert in that. And that's really noble, and I think that's really needed. But I think one of the things that is a downside of that is that you if it's not what you thought it was, or it doesn't participate in the process in the way that you think it does, or if it turns out that that disease mechanism as a whole is no longer of interest to the funding agencies, I've been able to work in a bunch of different areas, and that's because they're interesting questions in all fields. And if you are careful and you listen to these processes, you start to read, it turns out that you might be able to leverage prior learning to what you're studying right now.
Yeah.
I would say,
Yeah.
people's career, keep your eyes open and look broadly, because I think it's an important thing is to not silo yourself into a specific domain. I think the more integrative biologists, the forward-looking people are, the more they'll realize that these things you're studying are not unique to just what your organ system or disease process is.
Yeah.
The other thing that I do young researchers is endeavor that you're in is not a job, but part of who you are, because if you do and you find the thing that you love, it will bring you contentment and happiness, because every time you go to work every day, there's always something new to learn. There's always something new to discover. There's always little joys in every, and sometimes, there are things but there's learnings in there if you see it in that way, right? So if you change your perspective, see it as an opportunity to say, okay, well, this didn't go exactly the way I had hoped. The thing is that feedback is important to help tune you and make you a better person, and so make your grant a better grant. So ultimately, if you do that, then you'll actually see them at the surface, like they're painful, as something that will still enrich yourself. So passionate about what you do,
Right.
Make it a part of who you are. I think the other thing is that when you're thinking about your path as a researcher heading into medicine, you have a bunch of different ways that you could go. Some people go this traditional route that most think of as an academic route, as they say, where you complete a PhD and can you start you work towards your independence as an investigator, and those are very noble routes, and they do require a lot of investment of yourself and your time. And running a lab require a lot of work. And when I went back to academia, I didn't realize just how much work that was. But it's managing a small business, and students and staffing, achieving the overall goals, and so that can be daunting to some. But I think if you realize that up front, and you know what you're getting yourself into, then you'll probably be more successful than if you have this very Pollyanna view of the world that every grant gets funded, because they don't. Most grants have a low success rate. So that can be a little challenging. So
I love that, Paul, because you're preaching, curiosity and passion, and I completely agree with you, because, in my own career, on my worst day, I woke up, I was still excited. I did electrophysiology. I was still excited to hear the sounds of the neurons firing as I was recording, or whatever, or like looking at the scan of the brain, or looking at microscopy images. Like, even on your worst day, if you like and enjoy what you do. And if you're curious, I think that's pretty great advice. So thank you.
Yeah.
I can talk to you forever, and I have talked to you forever because I really love your philosophy and approach to science, and I love that you have this kind of holistic view, because I always held to this, like, the whole is greater than the sum of its parts.
Yeah.
And so as you mentioned, like, I think a lot of people end up during their PhDs and careers kind of specializing in these silos. But then it's such a treat to get to hear from someone like you, in my humble opinion, that does have some insight and has tapped into this kind of holistic view of normal biology, disease biology, how to measure it, and how to fix people and animals as they are aging or have conditions. But, given our time restrictions, I just have one last question. What are some of your future projects that we can look forward to? And, any other final thoughts that you might want to share with our audience today?
Yeah. So, for me, I'm personally excited to see what the next 10 years brings.
Okay.
Someone who's working in this translational space, medical research community at this preclinical stage, and also what's happening clinically is for next few years going to be really, really transformative. In particular, I think, with the advancement of our technologies that are in markers, imaging, pharmaceutical research, and the overall understanding of how these can be used and leveraged to help, I think, is going to be exciting. And in particular, we're, as I said, we're interested in a number of these areas as ways to treat patients, at least at the preclinical stage, animals as our patients, and then ultimately our plan is to translate many of these things into clinic. In particular, we have a number of key diagnostics in the pipeline that we're working on and have some really exciting results we hope to have out later this year at the very earliest stages of the disease. So diagnostics where we can see where AD is just starting right before you go into mild cognitive impairment. And I think that that's really an exciting area that we're really looking towards, because then that also gives us an opportunity to start to think about what sorts of interventions you might, apply. And this could include new therapies, but also could be repurposing existing drugs. And then the other piece that we're really excited about, I think, for ourselves is there are a number of new therapeutic approaches that are coming. Some of these are, as we were talking about, in the inflammation space. We're working obviously in that space as well. And we're also very excited about this notion of combinatorial therapy, because I think that actually will change the way that we see the disease. It's not just simply a single process. It's multiple processes all occurring at the same time. And, which of these do we need to turn off so that we can have some medical benefit, and which ones do we leave on so that you can still provide the normal function that tissue normally would do. And so that's an area that we're actively pursuing. And coupled to that, of course, is a diagnostic, this multimodal diagnostic approach, where we say, if we have a bunch of these measurements, can we put them together to see which would be the appropriate type of treatment that this person would benefit from most? And can we actually detect them at a stage when it would be most beneficial? And so this is an area that we're actively working on and have put in grants for. And so we're pretty excited about what's coming over the next 10 years. I think it's going to be an exciting time. So that's what we're thinking about.
So one thing I will say, we will put a link to your lab in the show notes, so people who are interested can also just keep on top of your research. You published quite a lot, and some fabulous articles that I've read and enjoyed myself through the various consortia that you're a part of as well. And so we'll make sure to put that in the show notes for anyone who wants to read more about your research and maybe even contact you. But thank you so much, call. It's been such a wonderful conversation. I always learn so much when I'm chatting with you, and I think, I'm very proud of this episode, if I may say that, because I think people will listen to it and come away with a much better understanding of biology and drug development and biomarkers and so many more things. So thank you again for your time. I know you're on vacation, so I really appreciate it.
Yeah, no, I really enjoyed myself. Thank you so much for having me. And as you said, I always enjoy chatting with you. And if listeners do have questions, they can feel free to reach out to me through the contacts that you'll provide. And so, it's been great. So thank you so much for having me.
Hey everyone, hope you enjoyed this episode of the No Brainer podcast, and see you at the next one.